Welcome. Thank you for joining us here at the Z-Files, where we will start now by asking you to imagine your body as a living machine, intricate and irreplaceable, yet vulnerable to failure. For decades, medical science has struggled to repair what breaks down inside us, relying on donors, waiting lists, and the fragile hope that a match will arrive in time. Now a new frontier is emerging, one where printers do not produce paper and ink, but living tissue, layer by careful layer, building the very organs we need to survive. Part 1. The crisis we face. Every day, somewhere in the world, someone dies waiting for a replacement body organ that never comes. The numbers tell a stark story. In the United States alone, more than 100,000 people currently wait for a transplant, and every 9 minutes another name is added to that list. 17 people die each day because the organ they need does not arrive. The gap between supply and demand has widened steadily for years, despite advances in transplant medicine and public awareness campaigns encouraging donation. The reasons are many. Donor organs must match the recipient in blood type, tissue compatibility, and size. They must be harvested quickly after death, transported under strict conditions, and implanted within hours before they deteriorate. Even when everything aligns, the recipient faces a lifetime of immunosuppressive drugs to prevent their body from rejecting the foreign tissue. These medications carry their own risks, including infection, kidney damage, and cancer. The system, for all its miracles, is built on scarcity and compromise. Kidneys are the most needed organs, followed by livers, hearts, and lungs. Some people wait years. Others never receive the call. Families watch loved ones weaken, tethered to dialysis machines or oxygen tanks, their lives measured in increments of declining function. Children grow up on waiting lists. The human toll is difficult to quantify fully, but it extends beyond the patients themselves to the families who care for them, the communities that lose them, and the healthcare system strained by chronic disease. Transplantation, when it works, is transformative. A new kidney can restore independence. A new heart can add decades. But the process depends entirely on the generosity of strangers and the timing of tragedy. It is a fragile system, and it cannot scale to meet the need. The world's population is aging, and with age comes organ failure. Diabetes, hypertension, and heart disease are rising globally. Demand will only grow. Traditional transplantation, no matter how refined, will never close the gap. Something else is required, something that does not depend on waiting for someone to die. That something may already be taking shape in laboratories around the world, where researchers are learning to build organs from scratch. The idea sounds like science fiction, but the science is real. Bioprinting, the process of using living cells as ink to construct tissue and organs layer by layer, has moved from theory to early practice. It is not yet ready to save lives on a large scale, but the foundations are in place. The question is no longer whether we can print living tissue, but how quickly we can make it functional, safe, and available to those who need it most. Let us begin by understanding what bioprinting is and how it works. Part 2. How bioprinting works. At its core, Bioprinting is a form of three-dimensional printing, but instead of plastic or metal, the printer uses living cells. The process begins with a digital model, usually derived from medical imaging such as CT or MRI scans. This model maps the structure of the organ or tissue to be created, providing a blueprint that guides the printer's movements. The printer itself resembles a conventional 3D printer, with a nozzle that moves along 3 axes, depositing material in precise patterns. The key difference lies in the material, known as bioink. Bioink is a mixture of living cells, often taken from the patient or a compatible donor, combined with a gel-like substance that provides structural support. This gel, called a hydrogel, mimics the extracellular matrix, the natural scaffolding that surrounds cells in the body. The extracellular matrix is not just filler. It provides signals that tell cells how to behave, when to divide, and how to organize themselves into functional tissue. Replicating this environment is one of the central challenges of bioprinting. The printing process itself is slow and meticulous. The printer deposits bioink layer by layer, building up the structure one thin sheet at a time. Each layer must align perfectly with the one below it. The cells must remain viable throughout, kept at the right temperature and supplied with nutrients. Once the printing is complete, the structure is placed in a bioreactor, a device that mimics the conditions inside the body. The bioreactor provides oxygen, nutrients, and mechanical stimulation, encouraging the cells to mature and integrate. Over days or weeks, the printed tissue begins to resemble the real thing. Cells form connections, blood vessels start to grow, and the tissue takes on the properties it will need to function. This maturation phase is critical. A freshly printed structure is fragile and immature. It must be nurtured carefully, monitored for signs of failure, and adjusted as needed. So far, researchers have successfully printed simple tissues. Skin is one of the earliest and most straightforward. It consists of layers of cells with relatively simple organization, making it an ideal candidate for bioprinting. Printed skin has been used in burn treatment and wound healing, providing a scaffold that helps the body repair itself. Cartilage is another success story. It lacks blood vessels, which simplifies the printing process considerably. Printed cartilage has been tested for joint repair and reconstructive surgery, with promising results. Bone tissue has also been printed, though integrating it with the body's own bone remains a challenge. These are not yet complete organs, but they represent important steps. They prove that living tissue can be printed, that it can survive and function, and that it can integrate with the body under the right conditions. The next step is more complex. Organs like kidneys, livers, and hearts are not simple sheets of cells but intricate structures with multiple cell types, branching blood vessels, and precise internal architecture. Printing them will require advances in resolution, speed, and our understanding of how cells self-organize. But the basic tools are in place, and progress is accelerating. Soon, we will explore what these advances might make possible. Part 3: Beyond Replacement. The promise of bioprinting extends far beyond simply replacing damaged organs. If we can print living tissue tailored to an individual, we open possibilities that traditional transplantation cannot match. One of the most significant is immunological compatibility. When an organ is printed using a patient's own cells, the risk of rejection vanishes. The body recognizes the tissue as its own. There is no need for lifelong immunosuppression, no gradual damage from anti-rejection drugs, no constant vigilance for signs of failure. The organ is a genetic match, identical in every relevant way to the tissue it replaces. This alone would transform transplantation, eliminating one of its greatest burdens and extending the lifespan and quality of life for recipients. But the potential goes further. If we can design organs, we might also improve them. Researchers are exploring ways to engineer tissue with enhanced properties, organs that resist disease, age more slowly, or perform better than their natural counterparts. A liver that metabolizes toxins more efficiently, a heart with stronger muscle fibers, a kidney that filters waste with greater precision. These are not fantasies but extensions of work already underway in genetic engineering and regenerative medicine. Bioprinting also offers hope for conditions that cannot be treated through transplantation. Some diseases damage tissue in ways that are too diffuse or too complex for a simple organ swap. Diabetes, for instance, results from the loss of insulin-producing cells in the pancreas. A full pancreas transplant is rarely performed, but printed clusters of insulin-producing cells, known as islets, could be implanted to restore function. Early trials have shown promise. Similarly, bioprinted patches of heart muscle might repair damage from a heart attack, restoring function without replacing the entire organ. Printed nerve tissue could help repair spinal cord injuries, reconnecting pathways that trauma has severed. The applications multiply as our understanding deepens. Each advance in printing technology, in cell biology, in materials science, opens new possibilities. Consider the economic implications as well. The cost of chronic disease is staggering. Dialysis alone costs tens of thousands of dollars per patient per year in the United States, and hundreds of thousands of people depend on it. A functional printed kidney, even if expensive to produce initially, could reduce those costs dramatically over a patient's lifetime. The same logic applies to heart failure, liver disease, and lung conditions. If bioprinting can scale, if production costs fall as the technology matures, it could ease the financial burden on healthcare systems worldwide. Access remains a critical question. New medical technologies often debut at prices only the wealthy can afford, creating disparities that persist for years or decades. Ensuring that bioprinted organs reach those who need them most, regardless of income or geography, will require deliberate policy choices, investment in infrastructure, and a commitment to equity. The technology itself is neutral, but the world it creates will depend on the decisions we make about how to deploy it. These possibilities rest on solving substantial technical challenges, challenges that occupy researchers today. We will turn to those next, and to the questions they raise about what it means to build a body. Part 4. The vascularization problem. One of the most stubborn obstacles in bioprinting is creating blood vessels. Every organ in the body depends on a network of arteries, veins, and capillaries to deliver oxygen and nutrients while removing waste. Without this network, cells suffocate and die within minutes. Simple tissues like skin can survive on diffusion alone, drawing oxygen from their surroundings, but anything thicker than a few millimeters needs its own blood supply. Printing the structure of an organ is one challenge. Printing the vasculature within it is another entirely. Blood vessels are not just tubes. They branch and subdivide in fractal patterns, growing smaller and more numerous as they penetrate deeper into tissue. The smallest capillaries are only a few micrometers wide, far thinner than the nozzles of current bioprinters can manage. The walls of these vessels consist of specialized cells arranged in precise layers, each layer performing a distinct function. Endothelial cells line the interior, regulating what passes between blood and tissue. Smooth muscle cells wrap around larger vessels, controlling diameter and blood pressure. The extracellular matrix provides strength and flexibility. Researchers have tried several approaches. One method prints channels into the tissue as it is built, leaving hollow spaces where blood vessels should be. After printing, endothelial cells are introduced, and they migrate into the channels, lining them and forming functional vessels. This works for larger vessels but struggles with the fine capillary networks that organs need. Another approach relies on the body's own angiogenesis, the natural process by which new blood vessels grow. Printed tissue is designed with chemical signals that encourage the body to extend its own vasculature into the implant. This can work, but it is slow, and the tissue may not survive long enough for the vessels to reach its core. A third strategy involves printing sacrificial materials that dissolve after the tissue is formed, leaving behind a network of channels. Each method has shown promise in laboratory conditions, but none has yet produced a fully vascularized organ ready for transplantation. The scale of the problem becomes clear when you consider a human kidney. It contains roughly 1 million nephrons, each a tiny filtering unit with its own blood supply. The total length of blood vessels in a single kidney exceeds several miles. Replicating this in a printer requires not just precision but also speed and coordination across multiple cell types. The printer must lay down endothelial cells, smooth muscle cells, and supporting matrix in the right places and the right proportions, all while keeping every cell alive. Progress is being made. Researchers have printed small sections of vascularized tissue that survive and function for days or weeks. They have developed bioinks that encourage vessel formation and bioreactors that stimulate growth. The gap is narrowing, but it remains the single largest technical barrier to printing complex organs. Solving vascularization will likely require a combination of approaches, along with advances in printing resolution and speed. When it is solved, the pace of progress will accelerate dramatically. Part 5. Nerves and signals. Blood vessels are not the only networks that organs need. Many tissues depend on nerves to function properly. Nerves carry signals from the brain and spinal cord, coordinating muscle contractions, regulating gland secretions, and providing sensory feedback. A heart without its nerve supply can still beat, driven by its own pacemaker cells, but it cannot adjust its rhythm in response to the body's demands. A printed bladder without nerves cannot sense when it is full or coordinate the muscles that control urination. The nervous system is woven through the body in patterns as intricate as the vascular system, and replicating it is equally challenging. Nerve cells, or neurons, extend long fibers called axons that can stretch from the spinal cord to the tips of the fingers or toes. These axons must connect to the right targets, forming synapses where signals pass from one cell to another. In development, this happens through a process of guided growth, where chemical signals direct axons to their destinations. Replicating this guidance in printed tissue is difficult. Current bioprinting methods can place neurons in the right general locations, but getting them to extend axons and form functional connections is another matter. Researchers are experimenting with materials that encourage axon growth, embedding chemical gradients in the printed tissue that guide nerve fibers toward their targets. Some have printed channels or scaffolds that physically direct axon extension. Others are using electrical stimulation to encourage neurons to mature and connect. Early results show that printed nerve tissue can conduct signals, at least over short distances, but integrating it with the body's existing nervous system remains largely uncharted territory. The challenge is compounded by the fact that different organs need different types of nerves. The autonomic nervous system, which operates largely outside conscious control, regulates heart rate, digestion, and other vital functions. The somatic nervous system controls voluntary movements and carries sensory information. Each type of nerve has distinct properties and requirements. For some printed organs, nerves may not be essential, at least initially. A kidney filters blood through passive processes that do not depend heavily on nerve input. A liver metabolizes substances based on chemical signals in the blood, not neural commands. But for organs like the heart, bladder, intestines, and lungs, nerve integration will be critical for full function. The good news is that the body has some capacity to re-innervate tissue on its own. When a transplanted organ is placed in the body, nerves from the surrounding tissue gradually grow into it over time. Printed organs might benefit from the same process, especially if they are designed with features that encourage nerve ingrowth. The timeline for full innervation could be months or even years, but if the organ functions adequately in the meantime, that may be acceptable. As with vascularization, the solution will likely involve a combination of printed structures, chemical signals, and the body's own regenerative abilities. These technical hurdles are formidable, but they are not insurmountable. Each year brings new tools and deeper understanding. Let us turn now to the questions that arise when we consider not just how to print organs, but where we must draw ethical lines when we do so. Part 6: Ethical Boundaries. As bioprinting advances, it raises questions that extend beyond the laboratory. If we can print organs, where do we draw the line? The technology is not limited to replacing damaged kidneys or hearts. In principle, it could be used to create tissue for cosmetic enhancement, to extend lifespan beyond natural limits, or even to modify the human body in ways that blur the boundaries of what we consider normal. Some of these possibilities are distant, but others are closer than we might think. Already, researchers have printed skin for burn victims and cartilage for reconstructive surgery. These applications are clearly therapeutic, aimed at restoring function and reducing suffering. But what about printing tissue to enhance appearance, to reverse aging, or to augment physical abilities? The line between therapy and enhancement is not always clear, and societies will need to decide where to place it. Regulation will play a critical role. Medical technologies are typically subject to rigorous testing and approval processes, but bioprinting is so new that regulatory frameworks are still being developed. Different countries may take different approaches, leading to disparities in access and standards of care. Another ethical concern is access and equity. If bioprinted organs become available, who will receive them first? Will they be distributed based on medical need, ability to pay, or some other criterion? History suggests that new medical technologies often debut at high cost, accessible primarily to the wealthy. Over time, prices may fall and access may broaden, but the intervening years can create significant inequalities. Ensuring that bioprinting benefits everyone, not just those who can afford it, will require intentional policy decisions, public investment, and perhaps new models of healthcare delivery. There is also the question of what counts as human. If we can print organs with enhanced capabilities, or if we begin to incorporate non-human cells or synthetic materials into printed tissue, at what point does the result cease to be fully human? This may seem abstract, but it has practical implications for identity, rights, and social cohesion. People have long grappled with the ethics of genetic modification, cloning, and other technologies that alter biology. Bioprinting adds another dimension to these debates. Religious and cultural perspectives vary widely. Some traditions emphasize the sanctity of the body as created by divine will, viewing modification as hubris or violation. Others see the relief of suffering as a moral imperative, embracing technologies that heal and restore. These perspectives will shape public acceptance and policy in different regions. Researchers and policymakers will need to engage with diverse communities, listening to concerns and incorporating values into the development and deployment of bioprinting. Transparency is essential. The public deserves to understand what bioprinting can and cannot do, what risks it carries, and what safeguards are in place. Overpromising or sensationalizing the technology can lead to unrealistic expectations and eventual backlash. Conversely, excessive caution or secrecy can fuel mistrust and resistance. Finding the right balance will require ongoing dialogue among scientists, ethicists, patients, and the broader public. These ethical questions do not have simple answers, but they must be addressed as the technology matures. The choices we make now will shape the future of bioprinting and its role in society. In the meantime, research continues, bringing us closer to the day when printed organs become a clinical reality. Part 7. Recent Breakthroughs. The past few years have brought remarkable progress in bioprinting, moving the field from proof of concept toward practical application. In early 2024, researchers at a consortium of universities announced they had successfully printed a functional miniature liver that survived for over a month in laboratory conditions. The liver, roughly the size of a walnut, contained multiple cell types arranged in the correct architecture, complete with bile ducts and a rudimentary vascular network. It metabolized drugs, produced proteins, and performed other essential liver functions at measurable levels. While far too small to replace a human liver, it demonstrated that complex organ printing is achievable with current technology. The same team is now working on scaling up the design, aiming for a quarter-sized liver that could support a patient while their own organ regenerates or while they wait for a traditional transplant. This approach, known as bridge therapy, could save lives even before full-scale organ printing becomes routine. Another significant advance came from Japan, where scientists printed sections of human heart tissue that contracted rhythmically for weeks. The tissue included cardiomyocytes, the muscle cells that generate the heartbeat, along with fibroblasts and endothelial cells that provide structure and blood supply. When subjected to electrical stimulation, the tissue responded appropriately, increasing its contraction rate in a manner similar to natural heart muscle. The researchers used a technique called aspiration-assisted bioprinting, which allows for finer control over cell placement and density. They are now exploring ways to integrate these printed patches with damaged hearts, potentially treating patients who have suffered severe heart attacks. Early animal trials are planned for later this year. Meanwhile, in the United States, a startup company received regulatory approval to begin human trials of printed skin grafts. The grafts consist of multiple layers including epidermis, dermis, and a vascular network, making them more sophisticated than earlier printed skin products. Initial results from burn patients show that the grafts integrate well, reducing scarring and improving healing times compared to traditional grafts. Progress has also been made in printing bone and cartilage. Researchers in South Korea printed a section of jawbone that was successfully implanted in a patient who had lost part of their jaw to cancer. The printed bone included a scaffold made of biodegradable polymers seeded with the patient's own bone cells. Over several months, the scaffold dissolved as new bone grew, eventually forming a functional replacement. The patient regained the ability to chew and speak normally. This represents one of the first cases where a printed structure fully replaced a significant portion of bone in a human. Each of these breakthroughs builds on decades of foundational research, but the pace is accelerating. Techniques that took years to develop are now being refined in months. Bioink formulations are improving, printing resolution is increasing, and our understanding of how to keep printed tissue alive and functional is deepening. Funding has increased as well, with governments and private investors recognizing the potential. The momentum suggests that the first printed organs suitable for transplantation may arrive within the next decade, perhaps sooner for simpler structures. The question is shifting from whether this is possible to how quickly it can be made safe, reliable, and accessible. Part 8. The Economics of Bioprinting. The financial implications of bioprinting extend across multiple dimensions, from research investment to manufacturing costs to the broader economic impact of reducing chronic disease. Developing the technology has required substantial funding. Government agencies, philanthropic organizations, and private companies have collectively invested billions of dollars over the past 2 decades. Much of this has gone toward basic research, understanding how cells behave in printed environments, developing suitable bioinks, and building the infrastructure needed for clinical trials. These are long-term investments with uncertain returns, the kind of spending that private markets often avoid. Public funding has been critical, particularly in the early stages when commercial viability was distant. As the technology matures, private investment is increasing. Startups focused on bioprinting have attracted venture capital, and established medical device companies are acquiring or partnering with them. The potential market is enormous, given the global burden of organ failure and chronic disease. Manufacturing costs remain high, but they are expected to fall as processes are refined and scaled. Currently, printing a single organ-sized structure can cost tens of thousands of dollars in materials alone, not counting the labor, equipment, and facility costs. Bioinks are expensive because they must be sterile, contain viable cells, and meet stringent quality standards. The printing process itself is slow, often taking hours or days for a single organ. Bioreactors and clean room facilities add further expense. However, these costs are likely to decrease significantly as the technology industrializes. Early computers filled entire rooms and cost millions. Today, far more powerful devices fit in our pockets and cost a few $100s. Bioprinting may follow a similar trajectory, though the comparison has limits given the biological complexity involved. Economies of scale will help. As production volumes increase, suppliers of bioinks and materials will be able to offer lower prices. Automation will reduce labor costs. Improved designs will shorten printing times. The economic benefits extend beyond the direct costs of producing organs. Consider the burden of chronic kidney disease, which affects roughly 10% of the global population to some degree. Advanced cases require dialysis, a process that costs healthcare systems billions annually. In the United States alone, Medicare spending on dialysis exceeds $30 billion per year. A reliable supply of printed kidneys could reduce this dramatically, freeing resources for other health priorities. Similar logic applies to heart failure, liver disease, and lung conditions. The productivity losses from chronic illness are also substantial. People unable to work due to organ failure represent lost economic output, reduced tax revenue, and increased demand for social support. Restoring them to health has multiplier effects throughout the economy. There are also opportunity costs to consider. Medical professionals who spend their time managing chronic disease could redirect their efforts if that disease becomes treatable through bioprinting. Research funds currently devoted to palliative care or incremental improvements in dialysis machines could be reallocated toward other challenges. None of this guarantees that bioprinting will be cost-effective in practice. Implementation will require new infrastructure, training for medical staff, regulatory oversight, and systems to manage quality and safety. Early adopters will face higher costs as they work through the learning curve. Insurance and reimbursement systems will need to adapt, determining how to pay for printed organs and under what circumstances. These transitions take time and involve complex negotiations among stakeholders. But the long-term economic case is compelling, particularly if the technology can reach a point where printed organs are comparable in cost to traditional transplants while eliminating waiting lists and rejection risks. As we consider these economics, it becomes clear that bioprinting is not just a medical innovation but a potential transformation of healthcare systems worldwide. The path forward depends on continued investment, careful stewardship, and a willingness to reimagine how we treat disease. Part 9. Engineering better organs. Once we master the ability to print functional organs, the next frontier is improving them. Natural organs evolved over millions of years to meet the demands of survival and reproduction, not necessarily to maximize lifespan or resist modern diseases. They are vulnerable to wear, disease, and the cumulative damage of aging. If we are building organs from the ground up, we have the opportunity to address these vulnerabilities. This is not science fiction. Researchers are already exploring ways to enhance printed tissue, making it more durable, more resistant to disease, or better suited to specific environments. One approach involves selecting the most robust cell lines for printing. Not all cells age at the same rate or respond to stress in the same way. By screening donor cells and choosing those with the greatest resilience, we can create organs that start with a biological advantage. Another strategy uses genetic modification to give printed cells enhanced capabilities. For instance, cells could be engineered to resist viral infection, reducing the risk of diseases like hepatitis in a printed liver. Cancer resistance is another target. Most organs accumulate mutations over time, some of which can lead to malignancy. Cells could be modified to include additional safeguards against uncontrolled growth, such as enhanced DNA repair mechanisms or fail-safe genes that trigger cell death if mutations accumulate. These modifications would need extensive testing to ensure they do not introduce new problems, but the principle is sound. Similarly, cells could be engineered to produce higher levels of protective proteins, such as antioxidants that neutralize the free radicals responsible for much cellular damage. An enhanced kidney might filter toxins more efficiently, extending its functional lifespan. An enhanced heart might resist the inflammatory processes that contribute to heart disease. These are incremental improvements, not radical transformations, but even small gains in durability could have significant impacts on patient outcomes. The concept raises questions about fairness and access. If enhanced organs become available, will they be reserved for those who can pay premium prices, or will they be distributed based on medical need? There is also the matter of unintended consequences. Evolution is a powerful optimizer, and natural organs reflect countless trade-offs refined over deep time. Changing one aspect of an organ might affect others in ways we do not anticipate. For example, increasing the metabolic rate of liver cells to improve toxin clearance might also increase their energy demands, requiring a more robust blood supply. Engineers and biologists will need to work closely, testing modifications thoroughly before they reach patients. Regulatory agencies will face difficult decisions about what counts as acceptable enhancement versus unacceptable risk. Despite these challenges, the potential is compelling. We accept that medical technology should do more than restore people to baseline health. Vaccines provide immunity beyond what nature offers. Antibiotics eliminate infections that would otherwise kill. Surgeries repair damage the body cannot heal on its own. Enhanced organs fit within this tradition, using technology to improve on biology where biology falls short. The key is to proceed thoughtfully, with rigorous testing, transparency about risks and benefits, and equitable access. If done well, engineering better organs could reduce the burden of disease, extend healthy lifespan, and improve quality of life for millions. The question is not whether we will pursue these enhancements, but how quickly and how carefully we move forward. As the science advances, society will need to grapple with what it means to improve on the human body and who gets to decide. Part 10. The role of stem cells. Much of bioprinting's promise depends on a particular type of cell, one with the remarkable ability to become almost any tissue in the body. These are stem cells, and they represent both the fuel and the foundation of regenerative medicine. Understanding their role is essential to understanding how bioprinting will transform healthcare. Stem cells exist in a state of potential. Unlike specialized cells such as neurons or muscle fibers, which have committed to a specific function, stem cells retain the flexibility to differentiate into multiple cell types. Embryonic stem cells, derived from early-stage embryos, can theoretically become any cell in the body. Adult stem cells, found in various tissues throughout life, have more limited potential but can still generate several related cell types. For bioprinting, the most promising candidates are induced pluripotent stem cells, or iPSCs. These are adult cells that have been reprogrammed to an embryonic-like state, regaining the ability to differentiate widely. The process involves introducing specific genes that reset the cell's identity, erasing its specialization and returning it to a more primitive state. The advantage of iPSCs is that they can be generated from a patient's own cells, typically skin or blood cells, through a laboratory process that takes several weeks. Once created, these stem cells can be expanded in culture, producing large populations, and then directed to differentiate into whatever cell type is needed for printing. A patient requiring a new kidney could provide a small skin sample. Researchers would reprogram some of those skin cells into iPSCs, coax them to become kidney cells, and use those cells as bioink to print a new organ. Because the cells originated from the patient, the resulting organ would be genetically identical, eliminating rejection risk. This personalized approach is one of bioprinting's most significant advantages over traditional transplantation. However, working with stem cells introduces complexity. Differentiation is not always complete or stable. Cells may retain some stem-like properties even after they are supposed to have specialized, and these residual stem cells can pose risks, including the potential to form tumors. Ensuring that printed tissue contains only fully differentiated, stable cells requires careful quality control. Researchers must verify that the cells have the right markers, behave appropriately in functional tests, and show no signs of abnormal growth. This adds time and expense to the printing process, but it is essential for safety. Another challenge is directing differentiation precisely. To print a kidney, you need not just kidney cells in general, but specific subtypes arranged in the correct architecture. A kidney contains epithelial cells lining the tubules, endothelial cells forming blood vessels, mesangial cells supporting the filtration structures, and others. Each type must be generated in the right proportions and placed in the right locations. Current protocols can produce most of these cell types from iPSCs, but the efficiency varies, and some cell types remain difficult to generate reliably. Advances in developmental biology are helping. By studying how organs form naturally during embryonic development, researchers are identifying the signals and sequences that guide stem cells towards specific fates. Replicating these signals in culture allows for more controlled and predictable differentiation. The long-term vision is to create libraries of differentiation protocols, standardized procedures that can turn iPSCs into any cell type on demand. With such libraries, bioprinting would become more routine, less dependent on trial and error, and more scalable. Stem cells also offer possibilities beyond printing entire organs. They could be used to generate patches of tissue for repair, or to create organoids, miniature simplified versions of organs used for research and drug testing. Organoids grown from patient-derived stem cells could allow doctors to test treatments in a personalized way, identifying which therapies are most likely to work for a specific individual. This precision medicine approach could improve outcomes and reduce the trial and error that currently characterizes much of clinical care. As stem cell science and bioprinting converge, the potential expands, limited primarily by our understanding of how to control cell behavior with precision. The cells themselves are ready. The question is whether we can master the orchestration required to turn them into functional, lasting organs. Part 11. Animal Models and Testing. Before any printed organ reaches a human patient, it must be tested extensively in animal models. This phase of research is critical, unglamorous, and fraught with ethical considerations of its own. Animal testing allows researchers to assess whether printed organs function in a living system, whether they integrate with surrounding tissue, and whether they trigger unexpected complications. The choice of animal model matters. Mice and rats are common in early research because they are inexpensive, reproduce quickly, and have well-understood biology. However, their small size limits what can be tested. A printed organ designed for humans is far too large to implant in a rodent. Researchers often use larger animals such as pigs, sheep, or non-human primates for later-stage testing. Pigs are particularly useful because their organs are similar in size and structure to human organs. Pig heart valves have been used in human transplants for decades. The anatomical and physiological similarities make pigs a good proxy for predicting how printed organs will behave in people. A typical testing protocol begins with acute studies, where the printed organ is implanted and monitored for hours or days. Researchers assess immediate function, looking for signs of blood flow, metabolic activity, and structural integrity. If the organ fails quickly, the team returns to the drawing board, adjusting the design or the printing process. If it survives, the next phase involves chronic studies, where animals live with the implanted organ for weeks or months. This reveals longer-term issues such as delayed rejection, degradation of the printed material, or failure of the organ to maintain function over time. Throughout these studies, animals are monitored closely. Blood tests track metabolic markers. Imaging studies visualize the organ in place. Behavioral observations ensure the animal is not suffering unduly. Ethical guidelines require that pain and distress be minimized, that animals be housed in appropriate conditions, and that the number of animals used be kept to the minimum necessary for reliable data. These are not abstract principles but enforceable standards overseen by institutional review boards and regulatory agencies. Despite these safeguards, animal testing remains controversial. Some argue that it is a necessary step in developing life-saving technologies, that the potential to alleviate human suffering justifies the use of animals in research, provided it is done humanely. Others contend that animal models are imperfect proxies, that results in animals do not always translate to humans, and that the ethical cost is too high. Both perspectives have merit. Animal models have limitations. Species differences in metabolism, immune response, and anatomy mean that success in animals does not guarantee success in humans. Drugs that work in mice often fail in clinical trials. Organs that function well in pigs may encounter unforeseen problems when implanted in people. These failures waste resources and can lead to harm if problems are not caught before human trials begin. Yet animal testing has also been essential to most major medical advances. Vaccines, antibiotics, surgical techniques, and transplantation itself all relied on animal research. Alternatives are emerging, including computer simulations, organoids, and tissue chips that mimic human physiology in miniature, but these are not yet sufficient to replace animal testing entirely. For bioprinting, the path forward likely involves a combination of approaches. Early-stage work can use computational models and in vitro systems to refine designs and identify problems. Mid-stage research moves to animal models to test function and safety in a living organism. Late-stage work transitions to human trials, starting with the smallest feasible interventions and scaling up as confidence grows. This stepwise approach balances the need for thorough testing with the imperative to minimize harm, both to animals and to the humans who will eventually receive printed organs. The process is slow, sometimes frustratingly so, but it exists for good reason. Lives depend on getting this right. As testing proceeds and data accumulate, we move closer to the moment when bioprinting leaves the laboratory and enters the clinic. That transition will require not just scientific validation but also regulatory approval, public trust, and healthcare systems prepared to deliver this new technology safely and equitably. Part 12. Regulatory Pathways. Every new medical technology must navigate a complex regulatory landscape before it can be used in patients. Bioprinting is no exception. In fact, because printed organs involve living cells, biological materials, and advanced manufacturing processes, they present unique challenges that existing regulatory frameworks were not designed to handle. Understanding these pathways is essential to understanding when and how bioprinted organs will become available. In the United States, the Food and Drug Administration oversees the approval of medical devices, drugs, and biological products. Printed organs potentially fall into all three categories. They are manufactured using devices, the bioprinters themselves. They consist of biological materials, the cells and bioinks, and they are intended to function like drugs or transplants, altering the body's physiology to treat disease. This ambiguity means that printed organs may be regulated as combination products, requiring approval under multiple sets of rules. The process typically begins with preclinical testing, the animal studies described earlier, which generate data on safety and function. If those data are promising, the developer can apply for permission to begin human trials. These trials follow a phased approach. Phase 1 involves a small number of patients and focuses primarily on safety. Researchers want to know whether the printed organ causes immediate harm, triggers immune responses, or fails in ways that endanger the patient. If Phase 1 succeeds, Phase 2 expands the trial to more patients and begins to assess efficacy. Does the printed organ actually work? Does it perform the functions it is supposed to? How does it compare to existing treatments? Phase 2 trials also gather more data on side effects and complications. If results remain positive, Phase 3 involves large-scale trials comparing the printed organ to the current standard of care. These trials can take years and involve hundreds or thousands of patients. Only after successful Phase 3 trials can the developer apply for full regulatory approval. Even then, approval may come with conditions, such as requirements for ongoing monitoring or restrictions on which patients can receive the treatment. Post-market surveillance continues indefinitely, tracking outcomes and identifying rare complications that might not appear in trials. This process is rigorous and time-consuming by design. The history of medicine includes cautionary tales of technologies rushed to market that caused unforeseen harm. Thalidomide, a drug prescribed for morning sickness in the late 1950s, caused severe birth defects because it had not been adequately tested in pregnant women. More recently, some medical devices have been recalled after patterns of failure emerged in real-world use. Regulators aim to prevent such outcomes by requiring extensive evidence before approval. However, this caution comes at a cost. Patients who could benefit from new treatments must wait years for approval, and some die while waiting. Balancing safety and access is one of the central dilemmas of medical regulation. For bioprinting, the challenge is compounded by the technology's novelty. Regulators lack established precedents. They must develop new guidelines, often in consultation with researchers, ethicists, and patient advocates. Different countries take different approaches. The European Union, Japan, and other regions have their own regulatory agencies with distinct requirements. A product approved in one country may face years of additional scrutiny elsewhere. Some developers pursue approval in multiple jurisdictions simultaneously, but this multiplies the complexity and cost. Harmonizing regulations internationally could streamline the process, but achieving consensus is difficult given differing legal traditions, risk tolerance, and healthcare priorities. Another complication is the personalized nature of many printed organs. Traditional drugs are manufactured in large batches, each batch identical to the next. Regulators can test samples from a batch and approve the batch as a whole. Printed organs made from a patient's own cells are unique. Each one is a batch of one. How do you regulate something that is never exactly repeated? Regulators are grappling with this question, exploring frameworks that approve the manufacturing process rather than the individual product. If the process is validated and controlled, if quality standards are met at each step, then each organ produced by that process can be presumed safe and effective. This approach is used in some forms of cell therapy, but applying it to bioprinting will require adaptation and innovation. Despite these hurdles, progress is being made. Regulatory agencies are engaging with bioprinting researchers early, providing guidance and feedback that helps developers design trials likely to meet approval standards. Expedited pathways exist for breakthrough therapies, treatments that address serious conditions with no adequate alternatives. Printed organs could qualify, potentially shortening the timeline from lab to clinic. The regulatory journey is not an obstacle to be circumvented but a necessary step in ensuring that bioprinting fulfills its promise safely and responsibly. As we navigate these complexities, the vision of widely available printed organs draws closer, built on a foundation of science, ethics, and the painstaking work of ensuring that what we create truly helps those in need. Part 13. The Learning Curve. When bioprinted organs finally reach clinical use, the medical community will face a steep learning curve. Surgeons, nurses, transplant coordinators, and other healthcare professionals will need to master new techniques, adapt to unfamiliar materials, and develop protocols for managing patients who receive printed tissue. This transition will not happen overnight. It will require training, practice, and patience as the first generation of practitioners learns by doing. Traditional transplantation already demands specialized expertise. Surgeons must connect blood vessels with microscopic precision, ensuring that the donor organ receives adequate blood flow immediately upon implantation. They must work quickly, as organs deteriorate outside the body, and they must be prepared for complications such as bleeding, rejection, or primary graft failure. These skills are acquired over years of residency, fellowship, and supervised practice. Bioprinting adds layers of complexity. Printed organs may arrive at the operating room in fragile states, requiring careful handling and specific preparation. They may need to be conditioned in bioreactors immediately before implantation, ensuring that cells are adequately nourished and that tissue has reached the right level of maturity. Surgeons will need to understand the characteristics of printed tissue, which may differ from natural organs in texture, strength, or healing properties. Suturing printed material might require different techniques or materials to avoid tearing. Connecting printed vasculature to the patient's own blood vessels will demand precision and an understanding of how printed vessels behave under pressure. Postoperative care will also differ. Patients receiving printed organs made from their own cells may not need the same immunosuppression regimens as transplant recipients, but they will require monitoring for other potential complications. Printed tissue might degrade or fail to integrate in ways that natural transplants do not. Early detection of these problems will be critical, and healthcare teams will need protocols for identifying and addressing them. Training programs will emerge, likely starting at academic medical centers with close ties to bioprinting research. Surgeons and other specialists will observe procedures, practice on simulators or cadavers, and gradually take on more responsibility under supervision. Professional societies will develop guidelines and best practices, distilling the experience of early adopters into recommendations that others can follow. Mistakes will happen, as they do with any new medical procedure. Some will be minor, caught and corrected without lasting harm. Others may cause serious injury or death. These failures will be scrutinized, analyzed, and incorporated into training and protocols to prevent recurrence. The process is iterative, a cycle of practice, feedback, and refinement. Beyond technical skills, healthcare professionals will need to manage patient expectations. Bioprinting will arrive accompanied by hype and hope, fueled by media coverage and the understandable desperation of people suffering from organ failure. Patients may expect miracles, immediate cures, or outcomes that the technology cannot yet deliver. Clinicians will need to communicate clearly about risks, uncertainties, and realistic timelines for recovery. They will need to help patients make informed decisions about whether to pursue printed organs or stick with conventional treatments. This counseling role is as important as surgical skill, shaping how the technology is perceived and whether patients feel satisfied with the care they receive. Hospitals and healthcare systems will also face logistical challenges. Bioprinting facilities will need to be built or retrofitted, equipped with clean rooms, bioreactors, quality control laboratories, and storage for bioinks and cellular materials. Supply chains will need to be established, ensuring reliable delivery of the raw materials required for printing. Insurance and billing systems will need to adapt, determining how to classify and reimburse printed organs. All of this requires investment, planning, and coordination across multiple stakeholders. The learning curve will be steepest in the early years, as the first few hundred or thousand patients receive printed organs and the medical community learns what works and what does not. Over time, as experience accumulates and techniques are standardized, the process will become more routine. What once seemed extraordinary will become ordinary, integrated into the fabric of healthcare the way kidney dialysis, heart surgery, and other once revolutionary treatments have been. The transition will test the resilience and adaptability of medical institutions, but it will also offer an opportunity to improve patient outcomes and reduce suffering on a scale that few technologies can match. Part 14: Global Disparities. Bioprinting has the potential to reduce health disparities by eliminating organ shortages, but it could also deepen inequalities if access is concentrated in wealthy nations or among affluent individuals. The technology's global distribution will depend on decisions made now, during its formative years, about research priorities, intellectual property, manufacturing infrastructure, and international cooperation. Currently, bioprinting research is concentrated in a handful of countries, primarily the United States, Japan, South Korea, China, and several European nations. These regions have the funding, technical expertise, and regulatory infrastructure to support advanced biomedical research. As a result, they will likely be the first to bring printed organs to clinical use. Patients in these countries will benefit earliest, while those in lower-income regions wait, potentially for years or decades. This pattern is familiar. New medical technologies often debut in high-income settings, where hospitals have the resources to adopt them and patients have the insurance or wealth to pay for them. Over time, costs may fall and access may broaden, but the lag can be substantial. Consider the history of antiretroviral therapy for HIV. Effective drugs became available in wealthy countries in the mid-1990s, transforming HIV from a death sentence into a manageable chronic condition. Yet millions in sub-Saharan Africa and other low-income regions continued to die because they could not access these medications. It took years of advocacy, price reductions, and international aid programs to extend treatment globally. Even now, disparities persist. Bioprinting could follow a similar trajectory unless deliberate efforts are made to ensure equitable access. One challenge is cost. As discussed earlier, producing printed organs is expensive, and early adopters will pay premium prices. High-income countries can absorb these costs more easily, either through national health systems or private insurance. Low-income countries, already struggling to provide basic health care, will find it difficult to invest in bioprinting infrastructure. Even if the technology becomes cheaper over time, the initial gap could persist if intellectual property protections limit the ability of generic manufacturers to produce bioinks or printers at lower cost. Intellectual property is a contentious issue in global health. Patents and proprietary technologies incentivize innovation by allowing developers to recoup their investments, but they can also restrict access by keeping prices high and limiting who can manufacture products. Some advocate for open-source approaches to bioprinting, where designs, protocols, and materials are shared freely, enabling researchers and manufacturers worldwide to participate. Others argue that without the financial rewards provided by intellectual property, companies will be less willing to invest in the risky, expensive process of bringing bioprinted organs to market. Finding a balance between incentivizing innovation and ensuring access is a familiar challenge with no easy solution. International organizations such as the World Health Organization could play a role, facilitating technology transfer, coordinating research efforts, and advocating for policies that prioritize global health equity. Philanthropic organizations and governments could fund the establishment of bioprinting facilities in low- and middle-income countries, training local scientists and clinicians to produce and implant printed organs. Another dimension of disparity is within countries. Even in high-income nations, access to advanced medical treatments is not evenly distributed. People in rural areas, members of marginalized communities, and those without adequate insurance often receive lower quality care. Bioprinted organs, like other cutting-edge therapies, may initially be available only at specialized centers in major cities, leaving rural and underserved populations behind. Addressing these internal disparities requires health policies that prioritize equity, invest in infrastructure beyond urban centers, and ensure that insurance covers new treatments regardless of where patients live. Public awareness and advocacy will be important. If bioprinting is perceived as a luxury for the wealthy, it may face political opposition or public skepticism. If it is framed as a public good, a technology that can save lives and reduce suffering for everyone, it may attract broader support and funding. The narrative matters. shaping how societies choose to invest in and regulate bioprinting. The goal should be a future where bioprinted organs are as accessible as antibiotics or vaccines, available to anyone who needs them regardless of geography or income. Achieving that goal will require vision, commitment, and international cooperation on a scale that the global health community has managed only rarely. But the alternative, a world where bioprinting deepens existing inequalities, is one we should strive to avoid. Part 15. Defining Humanity. As bioprinting advances, it forces us to revisit fundamental questions about what it means to be human. If we can replace damaged organs with printed ones, enhance those organs beyond natural capabilities, or incorporate synthetic materials into our bodies, where do we draw the line between human and something else? These are not merely philosophical curiosities but practical concerns that will shape how bioprinting is regulated, accepted, and integrated into society. For most of human history, the body has been understood as a fixed inheritance, shaped by genetics and development, vulnerable to injury and disease but essentially unchanging in its fundamental nature. Modern medicine has already challenged this view. We replace hips with metal and plastic, implant pacemakers and cochlear devices, transfuse blood, and transplant organs from one person to another. Each innovation has required society to adjust its sense of what is natural, what is acceptable, and what it means to be human. Bioprinting continues this trajectory but accelerates it. A printed organ made from a patient's own cells is perhaps the least disruptive, simply a more sophisticated version of transplantation. But what about an organ printed from stem cells that have been genetically modified for enhanced function? What about organs that incorporate non-human cells, such as pig cells used to provide structural support or growth factors? Some researchers are exploring xenotransplantation, the use of animal tissues in humans, as a complement or alternative to bioprinting. If a printed organ includes both human and animal cells, does it remain fully human? Most people would likely say yes, but the question becomes more complicated as the proportion of non-human material increases. At what point does a chimera, an organism containing cells from multiple species, cease to be one species or the other? These questions matter because they affect identity, rights, and social belonging. Laws and ethical frameworks are built on assumptions about what constitutes a human being. If those assumptions are destabilized, the frameworks may need revision. Consider the legal status of personhood, which carries rights such as autonomy, dignity, and protection from harm. If bioprinting allows us to create beings that blur the line between human and non-human, how should those beings be classified? This is not an immediate concern, but it is a foreseeable one, and it warrants discussion before the technology outpaces our ethical preparedness. Religious traditions offer varied perspectives. Some emphasize the sanctity of the body as created by a divine being, viewing modification as a violation of sacred design. Others see the relief of suffering as a moral duty, embracing technologies that heal and restore. These perspectives are not necessarily incompatible, but they lead to different conclusions about what kinds of modifications are acceptable. A printed kidney that replaces a failed one might be universally welcomed, but an enhanced kidney that performs beyond natural capacity might be viewed with suspicion or rejection by some traditions. Engaging with these perspectives requires humility and a willingness to listen. Scientists and policymakers cannot simply impose their views but must work with communities to find common ground. Cultural differences also play a role. Western societies tend to emphasize individualism and personal autonomy, valuing the right of individuals to make decisions about their own bodies. Other cultures prioritize community and collective well-being, viewing individual choices as something to be negotiated within a social context. These differences will shape how bioprinting is adopted and regulated globally. There is also the matter of enhancement versus therapy. Most people agree that using bioprinting to replace a diseased organ is ethically acceptable, but opinions diverge when it comes to enhancement. If we can print organs that are better than natural ones, should we? Who decides what counts as better? Enhanced strength, longevity, or cognitive function might seem desirable, but they could also create new forms of inequality, dividing society into the enhanced and the unenhanced. These concerns echo debates around genetic engineering, performance-enhancing drugs, and other technologies that promise to improve the human condition. The risk is that enhancements become a tool of privilege, accessible only to those who can afford them, widening the gap between rich and poor. Some argue for regulations that limit enhancement, restricting bioprinting to therapeutic uses. Others contend that such restrictions are paternalistic, infringing on individual freedom. Both positions have merit, and the resolution will likely vary across societies and evolve over time. What remains constant is the need for ongoing dialogue, a conversation that includes scientists, ethicists, patients, religious leaders, and the broader public. Bioprinting is not just a technical achievement but a social one, and its success depends on building consensus about how it should be used and what values it should reflect. As we navigate these questions, we shape not only the future of medicine but the future of humanity itself. Part 16. The Promise of Personalized Medicine. Bioprinting represents a culmination of a broader shift in healthcare toward personalization, a move away from one-size-fits-all treatments toward therapies tailored to individual patients. This shift has been building for decades, driven by advances in genetics, molecular biology, and data science, but bioprinting may be its most dramatic expression. The ability to create organs matched precisely to a patient's biology could transform how we think about disease and treatment. Traditional medicine operates on population averages. Drugs are tested in clinical trials involving hundreds or thousands of people, and if the average outcome is positive, the drug is approved for general use. But individuals vary. What works for most may fail for some, or cause side effects that others do not experience. Genetics, environment, lifestyle, and the unique history of each person's body all influence how they respond to treatment. Personalized medicine aims to account for this variation, using information about an individual to guide decisions about which treatments to use, at what doses, and in what combinations. Bioprinting takes this concept further by creating not just personalized treatment plans but personalized organs. An organ printed from a patient's own cells is inherently matched to their biology, eliminating the risk of immune rejection and the need for lifelong immunosuppression. This alone is a significant advance, but the possibilities extend beyond compatibility. Imagine a patient with liver disease caused by a genetic mutation that impairs the liver's ability to metabolize certain toxins. A printed liver could be engineered to correct that mutation, replacing the defective gene with a functional copy. The patient would receive not just a new liver but a better liver, one that addresses the root cause of their disease. Similarly, a patient with heart failure might receive a printed heart customized to their body size, blood pressure, and activity level, optimized for their specific physiology. These are not distant fantasies but logical extensions of capabilities that already exist in research settings. Gene editing tools such as CRISPR allow scientists to modify DNA with precision. Stem cell technology allows those modifications to be incorporated into cells that can then be expanded and printed into tissue. The challenge is integrating these tools into clinical practice, ensuring that modifications are safe, effective, and do not introduce unintended consequences. Personalized organs could also be used for testing. Before a patient undergoes treatment with a new drug or procedure, doctors could print a miniature version of the patient's organ, an organoid, and test the treatment on it. This would reveal how the patient is likely to respond, allowing doctors to adjust the approach before committing to it in the patient's body. This concept, sometimes called a digital twin or organ avatar, is already being explored in cancer treatment, where organoids grown from a patient's tumor are used to screen chemotherapy drugs. The approach could be extended to other conditions, providing a personalized testing platform that reduces trial and error. The economic implications are significant. Personalized medicine is often assumed to be more expensive than conventional approaches, but that is not necessarily true. If personalization allows doctors to choose the right treatment the first time, avoiding ineffective drugs and unnecessary side effects, it could reduce overall costs. A patient who receives a printed organ that functions well for decades may incur lower long-term costs than one who cycles through multiple treatments or experiences complications from rejection. However, realizing these benefits requires upfront investment. Generating iPSCs from a patient, differentiating them into the needed cell types, printing the organ, and maturing it in a bioreactor all take time and resources. Streamlining these processes, automating steps where possible, and developing standardized protocols will be essential to making personalized bioprinting economically viable. Insurance and reimbursement systems will need to adapt as well. Currently, most systems are designed around standardized procedures with predictable costs. Personalized treatments, with their variability and upfront expense, do not fit neatly into these frameworks. Policymakers and payers will need to develop new models that account for the long-term value of personalization, even if it costs more initially. There is also the question of how to handle the data generated by personalized medicine. Each patient's genetic information, medical history, and treatment outcomes contribute to a growing body of knowledge that can inform future care. Protecting patient privacy while enabling researchers to learn from these data is a delicate balance. Strong data governance frameworks, transparent consent processes, and robust security measures will be necessary. As bioprinting becomes more personalized, it will blur the line between medicine and manufacturing. Healthcare providers will not just administer treatments but will participate in their creation, working closely with bioprinting facilities to specify exactly what each patient needs. This integration of care and production represents a new model of medicine, one that is more responsive, more precise, and ultimately more effective. The promise is profound, but so is the responsibility to ensure that personalization benefits all patients, not just those with the means to access it. Part 17. Environmental Considerations. Bioprinting, for all its medical promise, is not without environmental impacts. The production of bioinks, the operation of bioprinters and bioreactors, and the disposal of biological waste all have ecological footprints that deserve attention. As the technology scales, these impacts will grow, and the field will need to address sustainability alongside efficacy and safety. Bioinks require raw materials, many of which are derived from biological sources. Collagen, a common component of bioinks, is often extracted from animal tissues such as bovine hides or fish scales. Hyaluronic acid, another frequent ingredient, is sourced from bacterial fermentation or animal tissues. Growing and processing these materials requires energy, water, and land, contributing to greenhouse gas emissions and resource consumption. Some researchers are exploring synthetic or plant-based alternatives that could reduce reliance on animal products, but these are still in early stages of development. The hydrogels used in bioinks must be biocompatible, biodegradable, and able to support cell growth, a demanding set of requirements that limits options. Bioprinters and bioreactors consume significant amounts of energy. Printing an organ can take hours or days, during which the printer's motors, heating elements, and control systems run continuously. Bioreactors maintain precise temperature, humidity, oxygen levels, and nutrient flow, often for weeks, all of which require electricity. As bioprinting facilities proliferate, their collective energy demand could become substantial. Powering these facilities with renewable energy sources such as solar or wind would mitigate their carbon footprint, but this requires infrastructure investment and policy support. Clean rooms, essential for preventing contamination during printing, also consume energy through constant air filtration and climate control. Water usage is another concern. Cell culture, a prerequisite for bioprinting, requires large volumes of sterile water for media preparation, washing, and maintaining bioreactor conditions. In regions where water is scarce, this could strain local resources. Recycling and purifying water within facilities could reduce consumption, but such systems add complexity and cost. Biological waste presents disposal challenges. Cells that do not survive printing, tissues that fail quality control, and materials used in the printing process must be handled carefully to prevent contamination and disease transmission. Incineration is a common disposal method for medical waste, but it releases carbon dioxide and potentially harmful pollutants. Autoclaving, which uses high-pressure steam to sterilize waste, is less polluting but still energy-intensive. Some facilities are exploring composting or other biological degradation methods for non-hazardous waste, reducing the volume that requires incineration. However, regulatory restrictions on handling biological materials limit these options. Packaging and transportation also contribute to the environmental footprint. Bioinks and cellular materials are often shipped in temperature-controlled containers with significant insulation and cooling mechanisms, many of which are single-use and end up in landfills. Developing reusable shipping systems or local production networks could reduce this waste, but coordinating such systems across the medical supply chain is complex. On the positive side, bioprinting could reduce some environmental impacts associated with traditional medicine. Organ transplantation requires the transportation of donor organs, often by air, which generates emissions. It also involves extensive use of medical facilities, surgical equipment, and pharmaceuticals, all of which have environmental costs. If bioprinting can produce organs locally, on demand, it could reduce some of these burdens. Additionally, by treating diseases more effectively and reducing the need for chronic interventions like dialysis, bioprinting could lower the overall environmental impact of healthcare. Dialysis centers consume large amounts of water and energy, and the dialysis process generates substantial waste. A successful printed kidney that eliminates the need for dialysis would offset its own environmental cost many times over. As the field matures, incorporating sustainability into research and development priorities will be important. Life cycle assessments, which quantify the environmental impact of a product from raw material extraction through disposal, could guide decisions about materials and processes. Collaboration with environmental scientists and engineers could identify opportunities to reduce waste, improve efficiency, and minimize harm. Bioprinting need not choose between medical innovation and environmental responsibility. With thoughtful design and a commitment to sustainability, it can advance both goals, contributing to a healthier population and a healthier planet. Part 18. The timeline ahead. Predicting when bioprinted organs will become widely available is difficult, but examining current progress and remaining challenges allows for reasonable estimates. The timeline varies depending on the complexity of the organ and the regulatory environment, but certain milestones are likely within the next decade. Simple tissues such as skin and cartilage are already approaching clinical readiness. Printed skin grafts have entered human trials, and regulatory approval for specific applications could come within the next few years. These products address real medical needs, particularly for burn victims and patients requiring reconstructive surgery, and they represent the first wave of bioprinting to reach patients. Cartilage for joint repair may follow a similar timeline, with initial approvals likely before 2030. These early successes will build confidence in the technology, demonstrate its safety and efficacy, and pave the way for more complex applications. Bone tissue is slightly further behind but progressing rapidly. Printed bone structures have been implanted in animals and in a limited number of human patients under compassionate use provisions. Full regulatory approval will require larger trials, but the path is clear. More complex organs face longer timelines. Kidneys, often cited as a priority given the enormous demand, present significant challenges due to their intricate internal structure and the need for extensive vascularization. However, research is advancing steadily. Several groups have printed kidney tissue that performs basic filtration functions in laboratory settings. Scaling this to a full-sized, fully functional kidney implantable in humans will likely take another decade at minimum. Optimistic estimates suggest the first human trials of printed kidneys could begin in the early 2030s, with broader availability by the late 2030s or early 2040s, assuming no major setbacks. Livers are similarly complex but may progress slightly faster because the liver has remarkable regenerative capacity. A partially functional printed liver could potentially support a patient while their own liver recovers, a less demanding application than full replacement. Bridge therapies using printed liver tissue could enter trials within the next 5 to 10 years. Hearts present unique challenges due to their mechanical demands. The heart must contract forcefully and rhythmically, pumping blood continuously for decades without rest. Printed heart tissue has been demonstrated to beat in laboratory conditions, but replicating the heart's full structure, including its valves, chambers, and conduction system, remains a formidable task. Initial applications may involve printed patches to repair damaged heart muscle rather than full heart replacement. These patches could enter trials within a decade. Lungs are among the most difficult organs to print due to their delicate gas exchange membranes and complex branching airways. Progress is slower here, and widespread clinical use is likely decades away. However, simplified lung tissue for toxicology testing or drug development may arrive sooner, contributing to research even if not directly treating patients. Regulatory approval processes will influence these timelines significantly. Expedited pathways for breakthrough therapies could accelerate progress, while stringent requirements for long-term safety data could delay. Public perception and acceptance will also play a role. If early applications succeed and gain public trust, subsequent approvals may proceed more smoothly. If high-profile failures occur, regulators and the public may become more cautious, slowing adoption. Funding is another variable. Sustained investment in bioprinting research, both public and private, will determine how quickly challenges are overcome. Economic downturns, shifts in political priorities, or competition from other medical technologies could divert resources and extend timelines. Despite these uncertainties, the trajectory is clear. Bioprinting is moving from laboratory curiosity to clinical reality. The question is not whether it will transform medicine, but when and how rapidly. Each breakthrough brings the technology closer to patients, and each patient who benefits strengthens the case for continued investment and development. Within the lifetime of many people alive today, bioprinted organs will likely become a standard option for treating organ failure, joining transplantation, dialysis, and other interventions as part of the medical toolkit. The pace of that transition depends on choices made now, on the commitment of researchers, policymakers, and societies to seeing this technology through its difficult middle stages to widespread deployment. The future is not fixed, but it is within reach, built one printed layer at a time. Part 19: A Vision of Healthcare Transformed. Imagine a future where organ failure is no longer a death sentence, where waiting lists have disappeared, and where patients receive organs perfectly matched to their bodies within weeks of diagnosis. This is the promise of bioprinting fully realized, a transformation of healthcare that extends beyond individual lives to reshape entire systems. The changes would ripple outward, touching economics, social structures, and the way we think about health and mortality. In this future, hospitals include bioprinting facilities alongside operating rooms and intensive care units. When a patient is diagnosed with kidney failure, liver disease, or heart damage, their cells are collected and sent to the printing lab. Within days, stem cells derived from those samples begin differentiating into the needed tissue types. The printing process begins, layer upon layer of living cells assembled according to a digital blueprint customized to the patient's anatomy. After maturation in a bioreactor, the organ is ready for implantation. The surgery proceeds much like a traditional transplant, but without the frantic race against time, without the uncertainty of donor availability, and without the burden of lifelong immunosuppression. Recovery is faster, complications are fewer, and outcomes are better. The economic implications are profound. Healthcare systems currently spend enormous sums managing chronic organ failure. In the United States alone, end-stage renal disease costs Medicare over $30 billion annually, most of it for dialysis. Cardiovascular disease, much of which involves heart failure, costs hundreds of billions when accounting for treatment, lost productivity, and premature death. If bioprinting can treat these conditions effectively, the savings could fund other health priorities, from preventive care to mental health services to research into diseases that currently lack good treatments. Insurance models would shift as well. Instead of paying for decades of dialysis or repeated hospitalizations for heart failure, insurers would cover the upfront cost of a printed organ and the associated surgery. This changes the calculus, potentially making bioprinting cost-effective even if the initial expense is high. Actuarial models would favor interventions that restore health over those that merely manage decline. Employment and productivity would benefit. People with organ failure often cannot work or work only part-time due to the demands of treatment and the limitations imposed by their condition. Restoring them to full health returns them to the workforce, benefiting not just the individuals but their employers, families, and communities. The social and psychological impacts are harder to quantify but no less real. Living with organ failure imposes constant stress, uncertainty, and dependence. Patients structure their lives around dialysis schedules, medication regimens, and doctor appointments. They live with the knowledge that their time may be limited, that their name on a waiting list may never be called. Removing this burden would improve quality of life immeasurably, allowing people to focus on work, relationships, and the activities that give life meaning. Families would benefit as well, freed from the strain of caregiving and the emotional toll of watching a loved one decline. Medical professionals, too, would experience this transformation. Nephrologists who have spent careers managing dialysis patients could redirect their expertise toward prevention and early intervention. Transplant surgeons would face less pressure and uncertainty, working with organs that are available on demand rather than dependent on tragedy. The shift would allow healthcare workers to focus more on healing and less on managing scarcity. research priorities would evolve. With organ replacement becoming routine, attention could turn to preventing organ failure in the first place, understanding the mechanisms of aging, and addressing diseases that currently lack effective treatments. The success of bioprinting could catalyze further advances in regenerative medicine, gene therapy, and other fields. This vision is not guaranteed. It depends on solving remaining technical challenges, establishing equitable access, navigating regulatory pathways, and maintaining public trust. It requires sustained investment, international cooperation, and a commitment to putting patient welfare above profit. But it is achievable. The science is sound, the progress is real, and the need is urgent. Every element of this future already exists in some form, in laboratories, in early trials, in regulatory discussions, and in the imaginations of researchers who see what is possible. What remains is to connect these elements, to scale them, and to deliver them to the people who need them. The transformation will not happen overnight, but it is underway, driven by thousands of researchers, clinicians, and advocates working toward a common goal. The future of healthcare is being printed, one cell at a time, and it holds the promise of longer, healthier lives for millions. Part 20. Your role in this future. You may wonder what role you can play in this transformation, whether you are a scientist, a healthcare worker, a policymaker, or simply someone interested in the future of medicine. The answer is that everyone has a part. Bioprinting will succeed or fail based not just on technical breakthroughs but on the choices that individuals and societies make about how to support, regulate, and deploy this technology. For researchers and students, the opportunities are vast. Bioprinting requires expertise across multiple disciplines, from cell biology and materials science to engineering and computer science. If you are drawn to this field, pursue education in these areas, seek out laboratories working on bioprinting, and contribute your skills to solving the challenges that remain. The field needs not just brilliant ideas but dedicated people willing to work through the difficult, unglamorous problems that stand between concept and clinical reality. Collaboration across disciplines is essential, so be willing to learn languages and methods outside your primary training. For healthcare professionals, staying informed about bioprinting will prepare you for the changes ahead. As the technology approaches clinical use, training opportunities will emerge. Engage with them. Advocate within your institutions for the resources and infrastructure needed to adopt bioprinting when it becomes available. Your voice matters in shaping how this technology is integrated into practice. For policymakers and regulators, the task is to balance innovation with safety, to create frameworks that allow bioprinting to advance while protecting patients from harm. This requires engagement with scientists, ethicists, and patient advocates, listening to diverse perspectives and building consensus. It also requires courage to approve new treatments when the evidence supports them, even if they challenge existing paradigms. Equitable access should be a priority, ensuring that bioprinting does not become a luxury for the wealthy but a resource available to all who need it. For advocates and patients, your experiences and voices are powerful. Patient advocacy has driven progress in many areas of medicine, from HIV treatment to rare diseases. By speaking out about the need for better treatments, by participating in clinical trials, and by holding institutions accountable, you can influence the direction and pace of bioprinting development. Organizations focused on organ donation and transplantation are natural allies, as bioprinting complements rather than replaces their missions. For the general public, education and engagement are key. Learn about bioprinting, understand its potential and its limitations, and participate in conversations about how it should be used. Support funding for biomedical research through your votes and advocacy. If you are able, consider donating to organizations that fund research into regenerative medicine and bioprinting. Public support translates into political will, which translates into funding and policy changes that enable progress. There are also practical steps anyone can take. Organ donation remains critical even as bioprinting develops, as the technology will not be available for all patients immediately. Registering as an organ donor costs nothing and could save lives. Supporting policies that increase access to healthcare ensures that when bioprinted organs become available, more people will be able to benefit. Engaging with ethical discussions about enhancement, equity, and the definition of humanity helps shape the social context in which bioprinting will exist. These may seem like small contributions, but they accumulate. Science and medicine are not separate from society but embedded within it, shaped by the values, priorities, and actions of ordinary people. The future of bioprinting will reflect the choices we make collectively, the research we fund, the regulations we enact, the patients we prioritize, and the values we uphold. It will reflect whether we see technology as a tool for the common good or as a commodity for the privileged. It will reflect whether we are willing to invest in long-term solutions or remain focused on short-term gains. The organ crisis we face today is solvable. The technology to solve it is emerging. What remains is the will to see it through, to ensure that the promise of bioprinting reaches those who need it most, and to build a future where organ failure no longer means waiting, suffering, and premature death. That future is not distant or abstract. It is being built now, in laboratories and hospitals, in regulatory agencies and policy discussions, and in the collective imagination of a global community working toward better health for all. Your role in this future begins with awareness, grows through engagement, and is realized through action. The story of bioprinting is still being written, and you are part of it.